Hey guys, welcome to my channel Life Sciences with SS. Today I am going to give a brief explanation about prion and prion related issues. The term prion refers to the abnormal pathogenic agent that are transmissible and are able to induce immune response. An abnormal folding of normal cellular prion protein that are most evidently found in brain. Previously, people think or understand that all diseases are either caused by any living organism, or if there is not the involvement of organism, there should be involvement of nucleic acid. But this concept is something different. In 1982, a scientist named Stanley B. Prusiner discovered that a protein particle is causing disease, and that disease was known as scrapie, which occurs in sheep. The cellular prion protein is found throughout the body, even in the healthy people, and their mode of action is very different because they don't have genetic material, they lack nucleic acid, and their mode of action is a present day challenge. This slide is showing the gain and loss of function hypothesis of prion disease pathogenesis. According to prevailing views, a pathogenic conformer can enter nervous system either through infection or as a result of conformational conversion of normal cellular prion protein. The abnormal conformer coops or changes normal molecule into an aggregate form by predominantly beta sheets containing abnormal protein at the expense of predominantly alpha helical cellular prion protein. The basic tenet or principle of gain of function hypothesis is that the infectious prion protein aggregation sensitizes neuron to program cell death and leads to prion disease. In turn, the loss of function hypothesis is based on the idea that loss of cellular prion protein leads to cell vulnerability and prion disease. The two hypotheses are mutually exclusive, and interdependence among infectious prion protein accumulation, cell death, and disease is still unsolved. Then this both diagram gives information about the cleavage. In first figure, when cellular prion protein is not cleaved by deletion or mutation, the prions elicit neurotoxicity. When alpha cleavage of cellular prion protein is blocked, the unstable prions tend to spontaneously aggregate to generate infectious prion protein. The generated infectious prion protein then affect the alpha cleavage of neighboring cellular prion protein to elicit neurotoxicity, or the process or the conversion process of cellular prion protein to infectious prion protein by itself transduces a toxic signal. A mutation or deletion may occur in a single allele or in a single cell. Then next slide is this slide is showing how cellular prion protein converts itself to infectious prion protein and then elicit aggregation and symptoms arouse. Endogenous cellular proteins when transforms itself into infectious prion protein, it gets free and free protein starts to coil and enclosed in a vesicle. Then it forms inclusion bodies. Inclusion means the cytoplasmic aggregation of stainable substances usually made up of protein. Then it forms amyloids or aggregates. The amyloids is the abnormality of plasma cells with abnormal light chain protein that is insoluble or structurally dominated by beta cell structure. Aggregates are cluster or massed bodies of unit or part somewhat loosely associated with one another. And then comes the step neurodegeneration. Neurodegeneration is the progressive dysfunction of specific function of neurons. Then only we can find desired symptoms. This slide is showing about how cellular prion protein changes itself to infectious prion protein, and there occurs no amino acid sequence change. Cellular prion protein is PK sensitive, which is protease kinase. PK stands for protease kinase. And here we can see cellular protein and infectious prion protein. And after circular dichroism, alpha is found to be 40 sorry, before circular dichroism, alpha is found to be 40% and 3%. And here circular dichroism occurs, and in infectious prion protein we can find alpha as 30% and beta as 45%. uh, then it is membrane bound form. Cellular prion protein is membrane bound form, and infectious prion protein is found in vesicles. Then comes next slide about the features of infectious prion particle. A single prion protein consists up to 50 amino acids, and after cleavage there are 22 amino acids, a signal peptide, and in cell surface there is in glycosylated and glycosyl phosphatidyl inositol, that is a GPI anchored protein, which is of 208 to 209 amino acid. They are involved in maintaining memory. Thing is that there are microglial cell patrolling throughout the body. Wherever they get damaged or bad nerve cell, they scavenge those nerve cells. And when a nerve cell dies, it can't function properly. It can't maintain our memory system, circadian rhythm, sleep-wake cycle, immune system, and many other symptoms arouse after that. In our body, some protein makes small adjustment in order to achieve best or desired performance, and this is all about the fine-tune process of normal protein. Then prion protein are not immunogen, and it does not easy to decompose biologically. They are not immunogen because they don't evoke immune system, because prion protein are body's own protein. They don't have any sequence change in amino acid. They just have a change in folding structure. They are just misfolded or abnormally folded one, and they are very stable form. They can't be degraded in high temperature or radiation that all other living organism can die in that temperature. But these prion protein are very much stable and very much resistant to any form of digestion, and they have ability to convert normal protein to abnormal ones, because they have ability to make bond with normal protein and convert them to misfolded one. And binding changes the conformation of normal protein, and they have affinity towards normal protein, and their bonding procedure is very strong. And they are not typical prokaryotic organism. We can't say that there is any organism in prion, because prions are not organism. They are only a protein particle, infectious protein particle. Then about this slide, this shows about the cleavage process and how they become infectious. In figure A, full length cellular prion protein has a function so that it is immediately cleaved at alpha cleavage site after activation. In second figure B, either N1 or C1 is an active form so that it is cleaved at alpha cleavage site to activate it. In third figure, cellular prion protein with mutation is not completely cleaved, so that the uncleavable prion shows some residual toxicity. Here we can see. And in last figure D, spontaneously generated cellular prion protein on one gene or derived from single cell blocks alpha cleavage of the remaining infectious prion protein. On one gene or derived from single cell blocks the alpha cleavage of remaining cellular prion protein, resulting in neurodegeneration. And infectious prion also block the alpha cleavage of host cellular prion proteins. And this slide is all about the infectious protein particle in various organism, in which organism we found which kind of prion disease. In animals, in like sheep and goat, we found scrappy, and transmissible milk encephalopathy is TME. This is one kind of prion disease. And then chronic wasting disease, CWD of mule bear, and bovine spongiform encephalopathy in cattle, we can call this as mad cow disease, and this is the most familiar prion disease which we can found in cow, cattle, etc. And in human, the most found prion disease is Creutzfeldt-Jakob disease, which you can say CJD, and Gertzmann-Straussler and Schrenker syndrome, GSS, and Kuru fatal familial insomnia, FFI. This Creutzfeldt-Jakob disease is mostly found in humans in various cases, majority of cases, and these are all about the prion disease in various organisms. And about spread from animal to animal, this prion protein can only infect animal to animal if they practice cannibalism, or if any human is having the meat of any infected cattle or any infected animal, then the human can be considered as infected. And this infection only concentrated in brain, no, in other parts of body it can't give so much effect, so much infection. It is only found in brain. And about the symptoms of prion disease, it involves rapidly developing dementia. Dementia is a group of thinking and social symptom that interferes with daily functioning, and difficulty in walking or changes in gait. Gait is the way in which someone walks. Then hallucination. Hallucination is the sensing something that seems to be real but are not. Then muscle stiffness, confusion, and fatigue. Fatigue is the feeling overtired with low energy and strong desire to sleep. And difficulty in speaking, we can say that the term is dysarthria for difficulty in speaking. These are the symptoms in prion disease. Then all about our conclusion. See, one thing I need to show you, this is the death rate in various age groups of male and female. We can see nearly age group from 70 to 79. We can see high death rate, and female have most high rate than male in various age groups. And middle, I mean in baby stage, no person seems to be infected, because this need to be minimum of 5 years to show symptoms. So about all conclusion, the spread from animals occurs through like feces, saliva, blood, urine, either through direct contact or environmental contamination, or also occurs from soil, food, water, etc. And still research is going on about this prion disease, and still various humans as well as animals die in this disease. No permanent cure is invented till now. So this is all about prion disease, and you can get this PDF on my Telegram channel named Life Sciences with SS. And if you have any doubt in this topic, you can comment me or can message me in Telegram account. And if you have any doubt in any other portion also, you can comment me. I will make videos with that. And if you like this session, please like, share and subscribe my channel. Goodbye.